At a fundamental evolutionary level, any multicellular organism relies on cooperative relationships between genetically identical cells. This cooperation creates a nutrient-rich environment amicable to life and ripe for exploitation. So there must be some sort of mechanism to identify and favor member cells while removing parasites. Plants and animals have developed similar innate immune mechanisms through conservation or coevolution to remedy this problem. These responses are well characterized in plants and animals, but in fungi, the innate immune mechanisms are largely unknown. Fungi use NLR and NLR-like receptors to recognize their own hyphal cells and hyphal fusion. And if fusion with non-self cells is attempted, regulated cell death may occur. But we don't know if these signaling pathways are similarly activated in some type of innate immune response to pathogens. My research uses two strains of the early divergent fungus, Rhizopus microsporus, as a model to investigate the mechanisms of innate immunity. The two strains are shown here. The host strain, RM13, houses endosymbiotic mycetohabitans SPP bacteria, B13. While these bacteria are extracted from the host and interacted with non-host strain, RM59, the non-host strain shows a reactive oxygen species response associated with innate immunity, indicated by the purple stain in this figure. Using this system allows me to compare an immune-activated state to a control state consisting of the same species of bacteria and fungus. My method is to extract the B13 bacteria from the host strain and interact them with the non-host strain in order to observe immune activation in the non-host strain in liquid culture, as seen in this diagram. RNA sequencing data, fluorescent staining, and flow cytometry and electron microscopy have all shown morphological changes in cell wall composition associated with the immune-activated state of the non-host strain interacted with bacteria. Specifically, there's an increase in exposed chitin and total chitin in immune activation, which appears to be associated with a thickening of the fungal cell wall. This may be an immune response analogous to callus deposition for cell wall reinforcement in plants. I plan to build on these results by comparing this fungal response to bacteria to the response to plant antimicrobial defenses to determine whether there are similar immune responses to different stresses in the fungus. This study is particularly relevant since our model fungus, Rhizopus microsporus, is a generalist post-harvest rot pathogen that causes significant food crop losses. An improved understanding of the fungal innate immune mechanism will have enormous implications on our understanding of the evolution of immunity and how these similar processes arose across the tree of life. On a more practical level, fungi play significant roles as food crops, natural decomposers, and plant and animal pathogens. An improved understanding of their immune defense will allow us to exploit these processes to support or suppress fungal development. Thank you.